So, good morning, everyone. Good afternoon, good evening, whatever timelines. Yes. So, welcome to the chapter Fundamental Unit of Life, Cell. We will be discussing about cell and tissues as well in this chapter. I'll be briefing you about the cell, not in much detail about the cell, I'm going to tell you, because it has been already done in detail many times. So, I will be briefing you. So, now let us start doing the fundamental basic units, that is what is the cell. Okay, many cells form tissues, and many tissues form organs, and many organs together form organ system, and all these organ systems will together make up an organism. Okay, so that is your hierarchy of the things, and we have many types of tissues. We are going to talk about it in today's class. Tissues, briefing about the cell. Now, let us talk about the cell. We do have two kinds of cells. One is your plant cell and animal cell. Okay, so what are the main differences? Let us first talk about the plant cell and animal cell. Animal cell differences. Very important question as per your CBSE or state board chapters. It is very important theory question here. All right. So yeah, according to the cell wall, if I have to talk about the cell wall is present. Cell wall is present and cell wall is absent in the animal cell. Cell wall is what? Is nothing but your outermost covering. Okay, so here is your cell, and this has got an outermost covering called as cell wall. This is my cell wall, which is present in the plant cell, whereas absent in the animal cell. And plastids do impart the color. Okay, plastids are present in the plant cell, whereas absent in animal cells. And when you come about the vacuoles, vacuole is nothing but that empty spaces. You have large empty spaces in plant cell, whereas small vacuoles, we have small vacuoles. Vacuole is nothing but that empty space wherein the food is being stored, because plants still will need a lot of space, because plants will prepare their own food and they store the food in those vacuoles as well, right? And vacuoles only one large central vacuole. Okay, one large central vacuole is present, whereas many small vacuoles are present in your animal cell. And then talking about the nucleus. Nucleus is generally eccentric, and here animal cell, the nucleus is centric. Centric, which is centrally present. Here it is eccentric, which means not exactly at the center. It can be somewhere here, or somewhere in this portion or somewhere. This can be your nucleus. This is your nucleus, need not be centrally present. Then you have something called as mitochondria. Mitochondria is nothing but your powerhouse of the cell, okay, where the energy powerhouse, okay. So where this is less in number for the plant cell, whereas more in number for the animal cell. And Golgi apparatus. Now, what are these Golgi apparatus? All these we are going to discuss now, right now. Okay, Golgi apparatus are many and scattered. And this Golgi apparatus is nothing but that packaging industry wherein your protein, food, everything is being packed and is being disposed to each and every part. Right. And whereas Golgi apparatus in the animal cell is only single, and it is near the nucleus. Near nucleus. Animal cell has got only one single Golgi apparatus, and that too near the nucleus. Golgi apparatus or Golgi body, we say, that is near the nucleus. Whereas in the plant cells you have many, because plant cells prepare their own food, right? So you need a lot of protein content dissipated there in small packets. Packaging industry, that is what is Golgi apparatus that would do. All right. So these are the major differences between your plant cell and animal cell. Now let us talk about the cell organelles, right? The compositions, functions of the cell organelles. Please do write everything as like this only in your tabular column in a very short and crisp note so that it will be easy for you to go with your, revision kind of things. Right. Now, let us talk about the compositions of cell, functions and all everything, right? Like, let us talk about composition, and you have functions here, and whichever is the component I am talking about, the component name, composition, and your yes. So, name of the components like cell wall. So, cell wall is that outermost covering that contains cellulose fibers that is made up of cellulose. Cellulose, and cellulose is nothing but your complex carbons, hydrocarbon or carbohydrates. I would say complex carbohydrates is nothing but your cellulose, and this is made of, cell wall is made of cellulose, which contains that thick fibers, cellulose fibrils we say. And what is the main function of it is to give the support and protection, because they are all thick fibers, na? So they give that protection, that they give that support to that body. And plasma membrane is nothing but it is having, it is made up of your lipids and fats. So this membrane, whatever you have this inner plasma membrane, this thick membrane is nothing but your plasma membrane, which is made up of lipids and fats. And this is a permeable membrane, I would say, wherein it selectively permits the molecules in and out of the cell. Selectively permits in and out of cell. And permits what? Permits the nutrition or any of the excretory wastes or water, whatever it is, whatever substances we are talking about, it selectively permits. It only permits those substances which is useful for the cell. And then you have nucleus, and nucleus is nothing but your main part in your cell, wherein it is responsible for the growth and reproduction, because during the time of reproduction, at that time when the cell is matured enough, the DNA in the nucleus that will split up, thereby it is mainly responsible for your growth. And nuclear envelope surroundings of you have nucleoplasm. Its composition is nucleoplasm. It is that thick jelly fluid in it, which is nucleoplasm, and it is made up of chromatin materials, which is nothing but the dense chromatid fibers and all, and nucleolus and nucleol. Okay, so these are the particles which it is made up of. Then number 4, it is ribosomes. Ribosomes is nothing but protein and RNA. Okay, the composition of ribosomes is proteins plus RNA. RNA stands for ribonucleic acid. Ribonucleic acid. RNA stands for your ribonucleic acid, and main function of it is protein synthesis. Protein synthesis. It is the protein synthesizing sites wherein you have something called as endoplasmic reticulum. Endoplasmic reticulum, which is just generally प्यार से उसको ER बुलाते हैं. ER, we have two kinds of ER. We have smooth endoplasmic reticulum and we have rough endoplasmic reticulum. We have two kinds. Okay, what does they both do? Now we have, I'm writing it here separately. Endoplasmic reticulum, we have SMOOTH. Smooth endoplasmic reticulum and rough endoplasmic reticulum. Smooth endoplasmic, they do not have ribosomes, do not have ribosomes, and uh, rough here, no, I'm writing it. Okay, yeah. Rough endoplasmic reticulum have ribosomes. And ribosomes, what are they? Ribosomes? Ribosomes are nothing but your protein synthesizing sites. They synthesize proteins, they generate proteins. Smooth ER do not have ribosomes. Do not have ribosomes. Then what is that they are responsible for? Smooth is responsible for membrane genesis. Membrane genesis. Matlab, in generating the membranes. So you have these, plasma membrane, nuclear membrane. Okay, so all these membranes, cell walls, you have all these membranes are generally, developed because of these lipids and fats. That is what smooth ER is made up of, that do not have ribosomes. It has got these lipids and fats responsible for generating membrane. Membrane genesis is the term we use. And rough endoplasmic reticulum, this is studded with ribosomes, and they synthesize proteins. Protein synthesizers, we say them as protein synthesizers. So this will be your, point number 5 here. Next, moving on, please write it down, everything in this chart like this only, so as to be easy on your studies while you study them. Next, then you have Golgi apparatus. We just now have discussed Golgi apparatus. It is the stack of membranous sacules, okay, wherein it is responsible for packaging the molecules. Process and packaging the molecules. And which kind of molecules? They are nothing but your glycoproteins. Glycoproteins. If I'm going a little fast, please pause it and write it down clearly. You can pause the video and write it down clearly and then again continue it. All right. So Golgi apparatus are nothing but your packaging industry. I would say this is packaging industry, wherein it packs all these proteins, all the nutrition, and then dissipates these packets to each and every corner of your cell wherever is necessary. And what do they pack? They pack glycoproteins. Okay, examples they are. Next, you have vacuoles. Vacuoles, or just now we have seen, they are the empty spaces, empty membranous spaces, and they are used for storage of substances. It can be proteins, it can be lipids, whatever it is storing. It is storage unit and matter. Okay, then you have lysosomes. Lysosomes are suicidal bags of the cell. We say suicidal, yes, you heard me right. Suicidal bags of the cell. What it does, it actually, contains, what is that it is having? It is having digestive enzymes. Digestive enzymes. So in case some part of the cell is unfunctional, is not working, okay, then what it does, it goes there, it liberates some digestive enzymes and it digests that particular part. And if once after this digestion, this part is being excreted out, your plasma membrane will open up and that part will be excreted out. Now, what if, the whole cell is disturbed, the whole cell is not functioning well? What if the whole cell is not going good on its functions? Then it bursts out. Lysosomes are those particles that will burst out in the cell with all the digestive enzymes, thereby digesting the whole cell, which means this whole cell is being digested now. So thereby everything is being digested, and that is what is called as suicide, right? So everything is vanished now. You don't have anything. So lysosomes are the suicidal bags of the cell, which has digestive enzymes in them, and they are definitely responsible for intracellular digestion. Intracellular digestion. Intracellular is within the cell. Digestion within the cell, responsible element here is your lysosomes. Then you have mitochondria, the powerhouse of the cell. Mitochondria is the powerhouse of the cell. It has got its own RNA, and it has got its own DNA, and ribosomes has got, it's a very important point, but please write it down carefully. Each and every point which I'm stating here is very, very, very important. Yeah. So mitochondria, it has got its own DNA and ribosomes. Okay. Why? Because see, this is the powerhouse of the cell. In case the power is not available with it, how does this work? So it has its own DNA, ribosomes, and ribosomes are nothing but your protein synthesizing sites. So proteins are being synthesized there, thereby liberating your energy there. And this is responsible for cellular respiration. Cellular respiration. Energy is always linked with respiration, isn't it? Respiration is nothing but your combustion in your physical terms, whereas respiration, we do say it in biological terms. Then, next, number 10, you have chloroplast. Chloroplasts. And chloroplasts and all, you see it in the plant cells, where in inner membrane are like, you have membranous sacs, chloroplasts, which have this grana and stroma. Grana it has got, and stroma it has got, all these stacking in these stacks kind of thing. You can see it in your book, in your textbooks. You have these pictures also, and they are used in photosynthesis. Used in photosynthesis, wherein chloroplast has got these, they are available in the chromoplast, and that has got the chlorophyll in it. Okay, chlorophyll is present here in the chloroplast. Each and everything has got its own, pigments and all. One of such pigment is chlorophyll present in chloroplast. So this is basically about your cell. Okay, so we have discussed about the organelles of the cell, and mainly about the differences between the plant cell and animal cell, right? Now let us come to tissues, the very, very, very important chapter, which is your tissues. So let's go with tissues right now. I hope you have written everything nicely, well in place. Right. Now we have to talk about plant tissues first. I'm talking about plant tissues first. You have something called as, see, you have two types here in plant tissues. One, meristematic tissues, meristematic tissues, and permanent tissues. And permanent tissues. Bache, please write it down, everything in this lot like this. Okay, and we have two kinds again. You have, according to the positions of them, we have apical meristematism, meristem, and then intercalary, intercalary meristem, and lateral meristem. So what do I mean by this? So here apical meristematism is, and any meristematic tissues, whatever these are all growth tissues responsible for growth of that particular plant. Okay, these are all your growth tissues responsible for growing. And as I say, these are also formed of cellulose, growth tissues formed of cellulose, and cellulose is nothing but your complex hydrocarbons or carbohydrates, I would say. Complex carbohydrates are nothing but your cellulose. This is nothing but a polymer of carbohydrates, Nana. It is huge molecules, these are all. And how are they? They are, rounded or polygon shaped they are, and they're compactly arranged. So not having so much of intercellular spaces, but yes, they do have some spaces in them, and they are capable of division, cell division. They are responsible for cell division, because we are talking about growth here. Meristematic tissues talks about growth here. The one of its important functions is cell division, capable of cell division and helps in growth. And they are localized in certain regions. Localized in certain regions. When I say localized, I am talking about these three things here, which are types of it, which are apical meristematism, then intercalary and lateral. Apical is nothing but, it is present in just the tips, in the tips. And what are those tips? If I have to talk about roots, root is one tip, then shoots, stem, branches, right? Stem, branches, wherever longitudinal growth is seen, longitudinal growth, I would say, because whatever the roots and all, and leaf tip also, like any tips, tip of the plant. Okay, you see your apical meristematism, and which is also called as primary growth. The primary growth in any plant is being seen in by the apical meristematic tissues. Shoot apical meristematism, and then root apical meristematism, roots and shoots also, and stem, branches also. The branches also do grow, right? So as the branches grow, that is also apical meristematism. And then you have intercalary meristematism. Intercalary, it lies at the internodes, at the nodes of your stems. Like if I have like this, okay, I have these are the roots and all I have. I'm very good at drawing, so please be careful with my drawing. Yeah. Wherever I have these branching developing, okay, branches coming out at these nodes, at these junction points is my intercalary, at these junction points. But as it is growing, here will be my apical at the tips. I hope you understand. Apical at the tips and intercalary at the nodes of the branches. Wherever I see my branching out things, I see definitely the apical meristematism, wherever your branches are coming out, the junction points, I would say, right? So wherever you see those junction points, there you see the intercalary meristematism. It is also helping in the longitudinal growth, also the primary growth of the plant. Then you have lateral meristematism. Lateral meristematism is the girth of the plant is being like it is girth of the plant. And what do I mean by girth of the plant? It is nothing but, see, the stem initially will be so patla sa, but now later on as the plant grows, this girth increases. The volume of it increases, like, right? Yes. So this is called as the girth. This is girth. Girth increases, and this is your secondary growth of the plant. Secondary growth, because once the shoot develops, once the roots develop, then the development of the girth is being observed. Once the plant holds in completely into the soil with developed roots, then you see this secondary growth there, which is increasing in their diameter of the stem. The diameter of the stem is increased. That is called as the girth increase, and which is being found in vascular bundles of dicot stems and all. Monocot, dicot, we have that differentiation as well. It is coming in now. Yeah. Then, so both are longitudinal growths, whereas, see, intercalary and apical and intercalary, both are my longitudinal growths. Longitudinal growth. Whereas lateral, lateral meristematism is growth in the diameter. Diametrical growth is observed here. I have also stated you the positions. The stem diametrically increases. Branch also, once the branch comes out at that node point, at that junction point, that is called as intercalary. Now, once this branch comes out, the branch also increases in the diameter, then that is called as your lateral meristematism, or which is increasing the girth of the plant. All right. So that is about your meristematic tissues. These are temporary tissues only. They are not permanent. Okay. So why are they not permanent? Why? Because they are responsible for the growth. So the plant will not grow on forever, right? So therefore, after a particular set of time, after a period of time, the plant stops growing. Then it turns into your permanent tissues. Then these all meristematic tissues will turn into the permanent tissues. So now please make a note of this. Plant tissues are mainly divided into two categories, which is meristematic tissues and permanent tissues. In the meristematism, we have apical, intercalary, and lateral. So now let us talk about the permanent tissues. Okay, I am moving on with my screen now. Please write it down properly. Okay, now coming up with my permanent tissues in detail. Yeah, permanent tissues. Now, in the permanent tissues, we have two kinds. One is simple permanent and the other is compound permanent. This is simple permanent tissues and the other is compound permanent tissues. Okay, let us first talk about simple permanent tissues. Simple permanent tissues, we have number one, parenchyma. Parenchyma. Number two, we have collenchyma. This is number two. And we have number three, sclerenchyma. Yeah, everywhere chyma. Okay. Yeah. Parenchyma, collenchyma, and sclerenchyma. Now, parenchyma is nothing but, so what is the differentiation between these three? So, permanent tissues are of two types, simple permanent and compound permanent. Compound permanent, we will talk about it later. Let us first finish this. Simple permanent tissues, which are parenchyma. These are all made of single type of cells. That's the reason they are called as simple permanent tissues. Single type of cells. Whatever type of cells which I'm talking about, this entire tissue is made up of the same kind of cells. What is a tissue? Tissue is nothing but a combination of cells, right? So all these cells are, together made up of, all these similar kind of cells will make up a tissue, which is called as your simple tissue or single type of tissue, simple permanent tissues. Now, if I have to talk about parenchyma, parenchyma, para is nothing but beside. Enchyma is fillings beside it, or you're just dumping in something. This is use it for storage purposes. Parenchyma main purpose is storage. It is a storage tissues wherein things are being stored there. So which kind of shape does that have? It is oval shaped or rounded or polygonal, whichever is the shape. And so usually they are loosely packed, because loosely packed, I'm writing that also, loosely packed, because something is getting stored into it. You have that, you should have that flexibility of allowing things into it, right? So that's the reason it has got thin walls also. Thin walls. If it has thick walls, things are not allowed inside. And parenchyma is a storage tissue. Plants often prepare its own food and gets stored there in the tissues there. So therefore, the food is being stored. You need thin walls for it, not thicker walls. And it's a live tissues, living tissue it is. It has got life in it, unlike sclerenchyma. Sclerenchyma is a dead tissue. We will write about that also. So this is living tissue. It is living cells. Then nature of it is cellulose. You have with pectin or lignin are present in it. There are large intercellular spaces. Yes, we do have. That's the reason I have written loosely packed. We have intercellular spaces, good amount of intercellular spaces, and they have large central vacuole in it. Large central vacuole. Yes, why not? Vacuole should be present because parenchyma is a storage tissue, and storage tissue, you need some space, right? So that's the reason it has been given like that. Then similarly, if I have to talk about the collenchyma, collenchyma is elongated, pointed ends. This is round, oval, and polygonal. Here it is elongated and with the pointed ends. Pointed ends. This is thick wall as compared to parenchyma, thick walls. It has got, and it is also living tissue only. It is also a live tissue, and it also has got pectin, pectinized corners. Pectin is nothing but, that glue kind of thing that gets, oozed out and makes the tissue harder. So that is what is pectin that is being released here, and little intercellular spaces, wherein parenchyma you have large, loosely packed annangada. So therefore you have more of intercellular spaces, more intercellular spaces here in parenchyma, less intercellular spaces in collenchyma. Wherein in sclerenchyma, we can see these are thick walls and dead tissues. Dead. They are all made up of dead cells. The bark of the tree, as you can see, this bark of the tree is made of sclerenchyma, bacha. And this is elongated fibers. Please, try to remember those shapes, oval, round, and polygonal shapes for parenchyma, elongated with pointed ends for collenchyma, and elongated fibers itself only for sclerenchyma. And the cell wall has got lignin. Lignin here, there, pectin here is lignin present in it, and there are practically no intracellular spaces, very, very, very less intercellular spaces. And that's the reason the bark of the tree is hard enough. It is not flexible, right? It is very hard, right? And where do you see the parenchyma tissues? Obviously, where do we see? In leaves, right? Leaf, because leaf is the one which will prepare the food, flowers, and fruits, definitely fruits, vegetables. Okay, everywhere you see these parenchyma tissues because these are the soft tissues. These are soft tissues because you have a lot of flexibility there because that is responsible for the storage. Allowing nature has to be there. So that's the reason it has got thin walls. So try to correlate to the function and its occurrence and its shape, right? And here they form collenchyma, they form the stem portion of it, and leaf stalk, a little bit thicker version of whatever we are talking about, leaf stalk of the leaf, and it's nothing but leaf may, like you have this middle rib, na? So that is also made up of collenchyma only. And leaf is made up of, yes, parenchymal tissues, wherein leaf stalk or this one's the pistil, all these are made up of flowers are made up of these collenchyma tissues. And here the sclerenchyma is nothing but the bark of the tree, which is generally like very hard bark of the trees. So this enables the plant to give that support. It gives the biggest support to the plant, to the plant or a tree, whatever you say. Okay. So these are the main functions, main occurrences. So function, its occurrence, and what kind of nature that it has. So all these three: permanent tissues, simple permanent, three types. So then comes up with your compound tissues, and in the compound tissues we have two things: xylem and phloem in the compound tissues. I hope this has been written well. Permanent tissues, you have two types. Okay. And two types, may one type we have just now discussed, simple permanent. Let us now discuss about the complex permanent tissues or compound permanent tissues. One xylem and the other phloem. And this is even important for your 10th graders. So xylem and phloem. Xylem, what do you mean by, this compound permanent tissues? These tissues are made up of not just the single type, not just the single type. It is made up of two or more kinds of cells making up one particular unit, two or more types making up one particular or turning up to do one particular function, and such tissues are called as compound tissues or complex tissues. And what are these? Like, because we have just now seen the simple tissues are made up of same type of cells. Whatever the shape is, every cell looks like the same. But here, xylem and phloem are compound or complex tissues which are having two or more types of cells, two or more types of cells together here. And so let us talk about the xylem here first. Xylem has got these four kinds, which are types of cells we have seen. So we have four types of cells together, four types. Xylem is formed out of four types of cells. Number one is your xylem tracheids, xylem tracheids, or tracheae, xylem vessels, VSSEL vessels. Then you have xylem parenchyma and xylem fibers. Okay, these are the four types of the cells which will form xylem tissue. And what does xylem do? Xylem has got one-way transfer of water and dissolved nutrients from the bottom part to the top part. This is xylem. Xylem, because where are you getting the nutrition from? You are obviously getting the nutrition from the soil. The plant is getting the nutrition from the soil. And how does it get the nutrition? Because of the dissolved nutritions in the water. Now, water gets into the plant, water gets into the roots of the plant because of osmosis. And what is osmosis? Cell to cell transfer of water from one cell to the other cell, water along with the dissolved nutrients. That is osmosis. And xylem performs osmosis, wherein one-way transfer of water and its dissolved nutrients from the roots till the tip of the plant. Each and every tip of the plant needs this nutrition to perform photosynthesis. So obviously, xylem is carrying all this to the topward end. Okay, and what are the functions of this? Xylem tracheids and vessels, both of these helps in long distance conduction. These help in long distance conduction of upward system of your dissolved nutrients and water. Definitely long distance, because it is sometimes the plants are very long enough, then you have to go till that point, then you should have a good amount of tracheids and vessels. And these are nothing but your tubule kind of things. These are the tubular kind of structures which will give the mechanical support, also gives you the support to the plant. Okay. It looks like this, like, the tube kind of fibers, wherein you have some outwards, wherein each and every branch or something here will also be going up with this, outlets. So, and then parenchyma, we just now have discussed. Parenchyma is all about storage, okay, which helps in storage of food. Okay, it helps in the storage of food and also lateral conduction. Conduction laterally. Laterally, matlab, it is one way towards the top end here. So laterally conduction of water and the dissolved minerals, not just water, not just the minerals, water and the dissolved minerals, because minerals are all dissolved in water, only then it is being transported. And obviously again it gives that support. Fiber gives the support, any kind of mechanical support is given by fibers, xylem fibers. Okay, they are dead and lignified. The xylem fibers are dead fibers and rest all are live. These are all live cells, whereas fibers are dead cells, wherein it provides that mechanical strength to it. Okay, and dead vibe, and it is being lignified also. Lignified. Cell walls you have here. So lignin is nothing but that cement kind of thing, which oozes out of the plant, making it hard on their outermost parts. So that is how your xylem fibers will be supporting them. And then now talking about the phloem part. So phloem goes with a two-way transfer. Now, what is xylem transporting? Xylem transports your food and the dissolved, not food, I'm sorry. Xylem transport the dissolved minerals and your water, right? Water and the dissolved minerals. Whereas phloem transports the food. Phloem transports food. So is that only one-way conduction? No, it is not one-way conduction. It is both the ways, down and upward. So why is it both words, upward and downward? How? Why? Reason? Because see, once the plant makes the food, transports food, and the food is being stored. Stored ante again it wants parenchyma also. Yes, it does have this parenchyma as well. Danik kuda four types unnai. It has got these four types, which are sieve tubes, companion cells, phloem parenchyma, and phloem fibers. Okay, sieve tubes and companion cells are just acting as your long distance conductance. Okay, long distance conductors. These tissues are nothing but these tubular kind of things, wherein they conduct the food, and food has to be conducted down and up. Like if I say carrot, okay, carrot, the main food is prepared by the plant in the leaves, right? Because leaves are the sites where photosynthesis take place. And photosynthesis, if I talk about, chlorophyll has to be present. So chlorophyll is present in the leaves and the green stems also. Yes, so there the food is being prepared and the food is being stored as you come down, sometimes as roots, sometimes as fruits, sometimes as flowers, sometimes as seeds. These are all the types wherein this food is being stored. Now I am talking about such a part wherein, example, root. Chalo. Okay, so carrot is nothing but the food which is being stored by the plant. Plant saves the food as a carrot in your root. Now, in the time of emergencies wherein the plant is not getting enough water or enough nutrients, it is not like any human body or any other animal body. The plant dies in just a minute. It is not that way. Plant will not die till a longer time, because the phloem conducts the food also from the bottom part to the topmost part, not just from the leaves to the bottom part, but also in case of any emergencies, in case of need, the food which is being stored inside the ground, as roots in the ground, that also will be brought back to the plant and gets utilized. So thereby phloem conducts food in both the ways, okay, downward and upward, both ways of conduction. And whereas your xylem is only one way of conduction, only from bottom to top, whereas phloem top to bottom and bottom to top. So it has got sieve tube cells, companion cells, which just stacks as your tracheids and vessels, which are your tubular in the shape, which will conduct long distance food transport there. And parenchyma is again the storage, as because we are talking about these plants there, so we definitely want this storage also here. And chief conducting tissue is your phloem for the food, whereas chief conducting tissues for the water and your nutrition is your xylem there. So this is about mainly about your differences also given, and it also gives support. See, the same for parenchyma and xylem parenchyma, phloem parenchyma, same. Xylem tissue fibers and phloem fibers functions are the same. So please note down the same functions for them as well. All right. So that ends up your tissues chapter for your plants. Plant tissues are done there. Then you have animal tissues. Okay. So I would be sharing another video for animal tissues here. Let us stop this plants tissues here on this. Okay, so please make a good note of it. And any doubts you have, you will let me know as you see the recording, the next class. You will let me know. All right. Chalo then. Happy learning to all of you. Bye bye. See you. Take care.